Hello boys, welcome back to another session of video tutorials. This session is meant for class 11th biology students and this is meant for 20th August 2020. Our memory helps us in reflecting upon what we have been discussing and studying. If you remember, we were discussing about various phases through which one glucose molecule undergoes oxidation and releases net 38 ATP molecules. And the various phases that a glucose molecule goes through is step number 1, glycolysis, breaking of glucose molecule. Lysis means breaking, glycolysis. Second, intermediate one, that is link reaction, where glycolysis results in breakdown of glucose and formation of two pyruvates or pyruvic acids. These two pyruvic acid molecules in the link reaction combine with NAD plus and coenzyme A in the presence of pyruvate dehydrogenase and Mg2 plus forms acetyl-CoA. Along with acetyl-CoA, energy-rich compound NADH is formed. So this is second. And the third phase is Krebs cycle or citric acid cycle, tricarboxylic acid cycle TCA, wherein 24 ATP molecules are produced per pyruvic acid. And remember, there are two pyruvic acids. One glucose molecule produces two pyruvic acids. And after this, the next phase is very important one, electron transport chain. As we said yesterday, the electron transport chain is the inner mitochondrial membrane has groups of protons H plus and electrons acceptors, proton acceptors and electron acceptors. So these groups are arranged in specific sequence, and this specific sequence is called ETC or ETS, electron transport system or electron transport chain. So basically in electron transport chain, it is the utilization of NADH and FADH that are formed in the Krebs cycle, as well as to make ATP synthesis. This is the main aim of ETC, electron transport chain. Utilization of NADH and FADH. And finally, to have this, there should be two things. One, electron flow should be there. Second, proton flow should be there. Third, proton gradient should be maintained. And of course, required enzyme should be there. Then only electron transport chain will happen. So as we explained yesterday, so in the inner membrane space, you can see 4H plus, 4H plus, 2H plus we have written. That means H plus means proton. There is a proton flow. And this proton flow will make the gradient. Down we have written electrochemical gradient. This plus 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 is because of H plus. And this minus is because of flow of electrons. And at the end of it, I want you to focus on last phase, that is F0, F1 particles of the mitochondria, and that happens in the inner mitochondrial membrane. And in matrix, there is conversion going on. NADH plus H plus is becoming NAD plus. That means NADH that is formed is being utilized. Now you can see this. And also FADH, that also is being used. And one molecule of water is formed. You can see here 2H plus plus half O2 becoming H2O. So for this to happen, various complexes are there. Like yesterday I said, there are totally 5 complexes in electron transport chain. One, it is called NADH dehydrogenase complex. This is complex number 1. As you are seeing here, complex number 1 is NADH dehydrogenase complex. So NADH is being broken to NAD plus, and the electrons are getting into FMN and then FES. And because of this, H plus is released and proton gradient will be created. And that electrons flow to ubiquinol. And then the electrons are flowing to ubiquinol hydrogenase, and then it enters into complex 3. Complex 3 is cytochrome BC1. So it consists of cytochrome B center and cytochrome C1. Electrons donated by COQH2 are transferred through cytochrome B, FES center and cytochrome C1. So in cytochrome C, which gets reduced, 4H plus are transferred from matrix to intermembrane space. So that's what you are seeing, 4H plus. And from there it is entering into complex 2, succinate dehydrogenase, wherein the succinate is forming fumarate, or succinic acid is becoming fumaric acid, and there FAD is produced. Remember, in a TCA cycle, and this FAD, FAS electron flow will be there, and that comes to cytochrome C. Now it enters into the electrons enter into complex 4th, that is cytochrome. Complex 4th is cytochrome C oxidase complex, and here what is happening? Again there is release of protons. So it includes cytochrome A, cytochrome A3, and 2 copper ions. You can see Cu, okay, copper ions named as A and B. Electrons are received from cytochrome C, which gets oxidized, and electrons are ultimately transferred to oxygen to form water. That's how the electrons are getting transferred to H plus protons, and protons and electrons together is making water formation. So in complex 4, it is the water formation that's important, and then the flow of hydrogens will be there, flow of protons rather, they enter into F0 F1 complex. And this in F0 F1 complex, the most important one is ATP synthase or mitochondrial ATPs. So ATPs are getting synthesized. So this is the crux of ETS, electron transport chain. And what is oxidative phosphorylation? Oxidative phosphorylation is the synthesis of energy-rich ATP molecules with the help of energy liberated during oxidation of reduced coenzymes. What are those reduced coenzymes? NADH and FADH2, produced in respiration. Hence it is different from phosphorylation, where light energy is utilized for the production of proton gradient that's required for phosphorylation. So the enzyme required for ATP synthesis is ATP synthase. ATP synthase is a must and becomes active only when there is proton gradient, that is H plus gradient. So the H plus should be high concentration in the outer chamber. That's why we are writing F0 plus plus plus. That is the rich proton gradient. And ATP synthase, that is complex 5, consists of 2 major components, as I said yesterday, that is F1, F0. The F1 headpiece is peripheral membrane protein complex, and it contains the site for synthesis of ATP from ADP plus inorganic phosphate, okay, adenosine diphosphate. F0 is an integral membrane, as all of you know, and it is a integral membrane of protein complex that forms a channel through which protons cross the inner membrane, as you are seeing 2H plus crossing. The passage of protons through this channel is coupled to catalytic site of the F1 component for the production of ATP. For each ATP produced, 2H plus 2 protons passes through F0. This is, you must remember this, many times this is asked in NEET exam. So how many protons pass through F0 from the intermembrane space to the matrix down the proton gradient? So you must remember 2H plus. And then higher proton concentration in the outer chamber causes protons to pass inwardly into the matrix or inner chamber through inner membrane. The protons are passed through special rotating channels in the region of F0 particles that reduces F1 particle to function as ATP synthase. So the creation of the enzyme ATP synthase is this way. The energy of proton gradient is used in attaching a phosphate radical to ADP. So only we are saying ADP plus inorganic phosphate forming ATP. So the method of forming ATP using electrochemical flow, that is proton flow, H plus flow through the membrane, is termed as chemisomatic ATP synthesis, or chemi-isomatic ATP synthesis, and is called chemi-isomatic theory, and it is proposed by Peter Mitchell. Now, what is the respiratory balance sheet that we have been talking? Okay, you can go through all this. I am interested in this. What is that? As we accounted in our previous class, glycolysis, direct synthesis, 2 molecules of NADH2, 1 molecule of NAD results in 3. So direct synthesis 2 ATP, 2 molecules of NADH, 2 into 3, 6. So 6 plus 2, 8. At the end of glycolysis, 8 ATP molecules are produced, right? And then link reaction, pyruvic acid to acetyl-CoA formation, 2 molecules of NADH, 2 into 3, so 6 ATPs. And citric acid cycle or TCA cycle, 6 NADH, 6 into 3, 18, 2 FADH, 1 FADH molecule is equal to 2 ATP molecules. So 2 into 2, 4, and direct synthesis of 2 ATP. So 18 plus 4, 22, 22 plus 2, 24. So in citric acid cycle, 24 ATP molecules are produced. So overall, 8 from glycolysis, 6 from the link reaction, and from Krebs citric acid cycle, 24. 24 plus 6, 30. 30 plus 8, 38. This is what we say. And then we say that this is amphibolic. Like, remember amphibia? 2 habitats, water, underwater, as well as on land. So here also we are using this word amphibolic pathway. Why are we calling this as amphibolic pathway? It is both anabolism as well as catabolism. Glucose is getting broken and also formed. Hence, since both the things are happening, breaking and making, we say amphibolic pathway. So glucose is favored substrate for respiration. We said yesterday, end of digestion, whatever we eat, finally what is formed? Glucose. So that glucose is used for respiration. Now this is what we say fats. Basically whatever food that we eat, it's of 3 types: fats, carbohydrates, proteins. So proteins become amino acids, and amino acids directly go and take part in the pyruvic acid. And fats become fatty acids and glycerols. Fatty acids, dihydroxyacetone phosphate, direct. And now carbohydrates, simple sugars, glucose, and glycolysis starts. Glucose 6-phosphate, fructose 1,6-phosphate, and breaking of that into 2, and then pyruvic acid formation, link reaction, acetyl-CoA, and Krebs cycle. So whatever we eat, finally it enters into Krebs cycle. So fats would need to be broken down into glycerol and fatty acids first. If fatty acids were to be respired, they would first be degraded to acetyl-CoA and enter the pathway. Glycerol would enter the pathway after being converted to PGAL, phosphoglyceraldehyde. The proteins would be degraded to proteases, and the individual amino acids, after deamination, depending on their structure, would enter the pathway at some stage within the Krebs cycle, and even as pyruvate or acetyl-CoA. Since respiration involves breakdown of substrates, respiratory process has traditionally been considered as catabolic process, and the respiratory pathway as catabolic pathway. But this is not completely true. When organisms needs to synthesize fatty acids, acetyl-CoA would be withdrawn from the respiratory pathway, and hence the respiratory pathway comes into the picture both during breakdown as well as synthesis of fatty acids. Similarly, during breakdown and synthesis of proteins too, respiratory intermediates form the link. So breaking down process within the living organism is catabolism, and synthesis is anabolism. Because the respiratory pathway is involved in both anabolism and catabolism, it would hence be better to consider the respiratory pathway as an amphibolic pathway. Now we come to the last topic of this chapter, respiratory quotient. It's called RQ. So what is the formula? RQ is equal to volume of CO2 liberated by volume of oxygen consumed. So like if you say, normally it gives, the value will be either 1, less than 1, 0, infinity. Now carbohydrates, if you take RQ is equal to 1. Why RQ is equal to 1? In carbohydrates, glucose plus oxygen, 6 CO2, 6 is 2, balanced reaction. So what is RQ? Volume of CO2. Volume of CO2 6 molecules. Volume of oxygen 6 molecules of oxygen. 6 CO2 by 6 O2 is equal to 1. So the RQ of carbohydrates is 1. What about fats and proteins? During aerobic respiration, that is germination of fatty acids, so this happens, tripalmitin. C51 H98 O6 plus 145 molecules of oxygen, 102 molecules of CO2, 98 molecules of water and energy. So RQ will be 102 CO2 by 145 O2. This becomes 0.71 for fats. And another one is that 63 molecules of CO2 is released divided by 77 molecules of oxygen, that is 0.9 for proteins. So it is RQ is always high for carbohydrates and low for fats. And then it is 0 when conversion of carbohydrates to organic acids takes place. Occurs when oxygen is utilized but CO2 is not evolved. In succulents where CO2 produced is utilized in carbon fixation. So it is glucose plus 3 O2. There is no release of oxygen. So that means 0 CO2. CO2 is not at this. See, you see the products, it is only malic acid plus water. So 0 CO2 by 3 molecules of. So that becomes 0 by something is 0. So this happens in succulents. Remember this for your NEET exam. And in organic acids, it is more than 1. During aerobic respiration, when organic acids are utilized as substrates, oxalic acid plus oxygen, 4 molecules of CO2. That means 4 CO2 plus 1 O2 is equal to 4. And carbohydrates, of course, it is infinity, because during anaerobic respiration where oxygen is not consumed but CO2 is released, that is alcoholic fermentation, glucose is becoming alcohol plus 2 CO2.